In this video, we're going to be talking about the next step of pathogenesis, the development of a disease, invasion. So invasion pathogens are going to move deeper down into the host tissues. In order to do this, they have to damage the host tissues, and that's what we're going to be focusing on in this video. So invasion, pathogens have to enter deeper down into the layers of the tissue, and this is going to require that the pathogen damage the tissue or somehow penetrate into the cells. And we're going to be talking about three ways that bacteria can do that: exoenzymes, toxins, and invasions. So exoenzymes, as you may be able to tell by the name, are enzymes that are released outside of the bacterial cell. So remember that word part, exo means outside. So exoenzymes are enzymes released outside of the cell. Two exoenzymes that are used for invasion are hyaluronidase and collagenase. So hyaluronic acid is the molecule that helps to hold nearby cells together. So remember that epithelial cells are going to form these tight connections in between each other in order to prevent bacteria from getting in between them. So bacteria are going to produce this hyaluronidase, which is going to break down this connection in between cells, so then bacteria can move in between them in order to reach the deeper layers of tissue. Epithelial cells are also going to have this basement membrane made up of connective tissue, which is going to be another barrier that they need to get through in order to cause an infection. This basement membrane made out of connective tissue is going to contain the protein collagen. So collagenase is going to break down that collagen in the connective tissue and allow it to move deeper down into the tissue. Another way of damaging tissues is through the production of toxins. So we have two main types of toxins: endotoxin and exotoxin. So remember, with endotoxin, there were four words that I always wanted you to put together: gram-negative, outer membrane, lipopolysaccharide, and endotoxin. Endotoxin is essentially just another term for lipopolysaccharide. So we find this in the cell wall of only gram-negative bacteria. It is called endotoxin because it's inside the cell wall. Exotoxin is going to be released outside of the cell. So endotoxin is a part of the cell, exotoxin is released outside of the cell. We are going to find endotoxins only in gram-negative cells because only gram-negative cells have that outer membrane where lipopolysaccharides are found, whereas exotoxins are going to be found in gram-negative and gram-positive cells. So comparing endotoxins to exotoxins, endotoxins found only in gram-negative cells. It's part of the cell wall. It's not specific to cell type. It has a relatively low toxicity, and it is heat resistant, meaning it doesn't break down in the presence of heat very well. Exotoxins, however, are found in both gram-negative and gram-positive. They get released outside of the cell. They are highly specific to cell type. They are highly toxic. Some of the most toxic substances on the planet are bacterial exotoxins, and they are heat sensitive for the most part. They are relatively easy to break down with heat. Both of them are going to allow the bacteria to damage the host tissues in order for them to invade deeper into the host. So endotoxin, again, this is essentially another term for LPS, lipopolysaccharide. We find it only in the outer membrane of gram-negative cells. Because it's part of the cell wall, it's only going to be released from the cell either when the cell dies, cell lysis, or when the cell divides, binary fission. There are two components to LPS: lipid A, which is the actual toxic component, and O antigen, which is the antigenic portion. So looking at the lipopolysaccharide right here, we have the lipid A here, and we have the O antigen, or the O polysaccharide, up here. So how does lipid A work as a virulence factor? So lipid A is a very strong activator of the immune system, and it triggers inflammation. This is actually beneficial to us when LPS is present in very low concentrations because it allows our immune system to recognize and respond to bacterial infections. So if there's a low concentration of LPS, our immune cells are going to recognize it. We're going to get a localized release of cytokines that trigger inflammation, and our immune cells are going to target and attack the bacteria, and we have a normal immune response. However, if there's a high concentration of LPS, then we are going to have the lipid A activate many immune cells, and instead of a localized release of cytokines, we get a systemic release of cytokines. So we get cytokines released all over the body. This is going to trigger a drop in blood pressure, which can lead to shock, organ failure, and eventually death, and we call this state endotoxemia. Exotoxins, remember these are found in both gram-positive and gram-negative cells. They are going to be released from the cell, and they are going to be highly specific to which cell they affect and how they affect that cell. Then they are going to be made up of proteins, which is why they're relatively heat sensitive. We have three main types of exotoxin: intracellular targeting toxins, cell membrane disrupting toxins, and superantigens. Intracellular targeting toxins, these are going to go inside of the cell, and they are going to target specific things inside of the cell. This is going to include what we call enterotoxins, which target intestinal cells, and neurotoxins, which target nerve cells. Then we have cell membrane disrupting toxins. These are going to attack the cell membrane, and these are going to include hemolysins, toxins that destroy red blood cells, and leukocidins, toxins that attack white blood cells. And then we have these superantigens. These superantigens trigger these non-specific immune responses, which can cause a lot of damage to the body. And this is going to include toxic shock syndrome toxin, a little bit redundant of a name. So cholera toxin, this is a toxin produced by the bacterium Vibrio cholerae. It is an intracellular targeting toxin that is an enterotoxin, which means that it targets intestinal cells. And what it does is once it gets inside of the cell, it's going to trigger that cell to release large amounts of electrolytes. When it releases those electrolytes, water is going to follow those electrolytes, and that is going to trigger lots of water being released inside of the intestinal tract, which is going to cause what's called rice water diarrhea. And you actually produce so much diarrhea that it can actually be fatal within hours of symptoms developing due to dehydration. This is going to both damage the host tissues and also help the bacteria to spread around, because Vibrio cholerae is going to be a waterborne pathogen that is spread through the oral fecal route. Botulinum toxin, this is going to be an intracellular targeting toxin that's a neurotoxin that's going to be produced by Clostridium botulinum. So what botulinum toxin does is it's going to target the neurons at the neuromuscular junction, which is how your nervous system controls your muscles. And what it does is it prevents the release of acetylcholine. And acetylcholine is the neurotransmitter that causes your muscles to contract. So if your muscles can't contract because acetylcholine isn't released, then they stay relaxed. It causes all of your muscles to relax, including your diaphragm, eventually causing you to suffocate. This is actually the active component of Botox, which is what causes your face to relax. Note, Botox also has some very important medical uses along with the cosmetic uses of Botox. It is the most toxic substance known, botulinum toxin. 1 gram is enough to kill 1 million people. It is super duper toxic. Kind of the opposite of botulinum toxin is tetanus toxin, which is produced by another member of the genus Clostridium, Clostridium tetani. This is another intracellular targeting toxin that's a neurotoxin, but this is going to cause your muscles to contract. So what it does is it's going to target these inhibitory interneurons and prevent them from releasing GABA and glycine, which are the signals for the muscles to relax. Since the muscles can't relax, they stay contracted, and it's going to lead to lockjaw, eventually paralysis of the diaphragm, leading to suffocation. There's this very characteristic arching of the back that is a sign of tetanus. Fortunately, we do have the tetanus vaccine, which is going to cause your body to produce antibodies that don't target the Clostridium tetani They actually target the tetanus toxin and prevent it from binding to your neurons and prevents it from causing the effects. Streptolysins, which are produced by Streptococcus pyogenes. These are cell membrane disrupting toxins. So this is specifically what's called a hemolysin. So it's going to target red blood cells. Remember that hemo word part means blood. So hemolysin, red blood cell destroying toxin. And how it works is it forms these pores inside of the red blood cells, which cause them to lyse. We classify streptolysins as either O streptolysins or S streptolysins, depending on whether or not they work in the presence of oxygen. So why, if you're a bacterium, do you want to kill red blood cells? Well, part of what bacteria need in order to reproduce is iron. So by killing these red blood cells, it's going to free up nutrients like iron for the bacteria to use to reproduce. Most bacteria, when they cause infection, cause what are called tissue infections. So they actually grow in between cells, not inside of the cells themselves. Some bacteria are able to cause intracellular infections. So they actually go inside the cell. In order to do this, they're going to produce these chemical signals called invasions. So invasions trigger the cell to actually take up the bacterial cell, so then it can go inside of the cell and cause an infection. So some notable pathogens that are going to cause intracellular infections include Listeria and Salmonella and EPEC. This is enteropathogenic E. coli, which is a strain of E. coli that's going to cause severe gastrointestinal symptoms. So this is showing specifically Listeria monocytogenes and how it causes an infection. So it produces these invasions which cause the cell to take up the bacteria, then it escapes the vesicle that it's taken up in, and then it's going to trigger the formation of what are called these actin tails, which you can see in this picture here. And what these do is they actually push the bacterial cells around inside of the cell, and they can actually push the bacteria from cell to cell. This way, the Listeria is able to bypass barriers inside of the body, like the placenta or the blood-brain barrier, which makes Listeria monocytogenes very dangerous, especially for pregnant women.